people will just trickle in as they come. My name is Jonathan Degner. I'm a forest research scientist with the BC Ministry of Forests. I'm here to moderate our first Q&A of the day for the Operational Tree Improvement 1 session. I'm here joined by our speakers, Tara Androschuk, Esteban Galeano, Dawei Loa, and UAU. Good morning, everyone. Do we have any questions yet? Not seeing any. So I'll start off with a question of my own. And that's a question for Tara. So this question around seed yields in lodgepole pine is a really important question that you're tackling, not just in Alberta, but that's one that we're dealing with in BC as well. And I find it quite interesting, the associations you're starting to see between seed yield and grafts versus seedling orchards. We're exclusively using grafted orchards here in BC, but are starting to move towards some seedling-based orchards. So that's going to be quite interesting for us. I was wondering if there were any substantial differences in remettent seedling age between those orchards, or if you could just kind of tell me the sequence in which they were established.
Yeah, so the B1 phase 1 orchard was established first. So that's the oldest orchard, and then that's like primarily seed origin, which has the fewest problems with generating seed. And then the other two orchards were established later, and they're primarily grafts, and they're the ones that are having the most difficulty with producing the seeds.
And what are the ages of those orchards presently?
Gosh, off the top of my head.
Sorry to put you on the spot there.
Yeah, I can't remember. I can look it up quickly.
Okay, we can circle back to that. I'm just curious. I know that orchard age, the dynamics change quite a bit, but we have some questions filtering in here. First off, we have a question for Esteban asking, how can industry directly use the information that you've learned so far? I guess we could sort of rephrase that as, are there maybe any sort of immediate implementations of your research that can be used?
Yes, Jonathan, and thanks for anonymous attendee. So not all of you may know, but Alberta has begun using molecular markers very recently. And it began with the RESTOR project a couple of years ago, like 5, 6, 7 years ago, for specifically genomic selection. In terms of pedigree reconstruction, pollen contamination, assessment, and affected population size using molecular markers, this is the first time. So I am answering the question right away. It is still like an unknown situation where this is the first work using the SNPs for a tree breeding program. It is a challenge for everyone, for the university to explain how it works, for the industry to understand the applications, and for the government to know if the regulations can or need the regulations need to be changed. So let's following the question, I think that it is a huge opportunity for the industry to get better gains, as we all know, against estimations using the co-ancestry or the gene matrix. And the good news is that we are doing this year and upcoming years other pedigree reconstructions for other tree improvement programs in Alberta. So we have different companies approaching the tree improvement lab, and we don't know the cost yet, but how long it will gonna take. But definitely, everyone seems to be open to talk, to negotiate, to understand, and particularly the government. The government has been a huge participant on this. First, to understand, and second, to be open in terms of accepting the use of the SNPs for pedigree reconstruction. For genomic selection, we are still ongoing, but pedigree reconstruction using SNPs seems to be good for a go. So I hope that I answered the question. It seems to be very, very a good opportunity for them, and it takes some time, you know, Jonathan, and the anonymous person who asked. It takes some time for everyone to understand and then to commit. But I think that we have done a tremendous job, everyone in the three different parts. So I am very, very positive. We need to be very positive and optimistic on this.
Thanks very much. We have many more questions for Esteban here. One that I was also quite curious about here is coming from Nick Ukrainitz. So in your pedigree reconstruction of the progeny trials, you found 30% pollen contamination. What was the experimental design of that progeny trial? Was it open pollinated in an orchard or polycrossed, or were these supposedly controlled crosses?
Yeah, I would like to answer very quickly, Haley Thomas. Could you explain how you calculated NE in your study using coancestry? We have three different types of contemporary assessment of NE, like with linkage disequilibrium, with heterozygote excess, and the coancestry. I use coancestry. Napoli Isabel was asking what is the minimum numbers of SNPs that will be required to monitor their seedlots. So for sure, we used all the SNPs available for the Jambusket chip, which is 5,000. But I took two different subsets, 2,000 SNPs randomly selected and other 2,000 SNPs randomly selected, and both worked perfectly. So I just kept any of those two subsets. And then, yes, Jonathan, just coming back to Nicholas. So first, the progeny tests are open pollinated trees, and definitely 30% is very distressing because it can be either errors when planting, when selecting seeds for growing the seedlings, or either not just when they were collected, they were coming, the open pollinated seeds were just with fathers that we didn't want to. So I don't know if I answered the question. Do you have a theory about?
Yeah, no, that's
okay.
That's a good answer.
Okay.
I think if it had been, for example, like a polymixed trial, that would be much more concerning. But if it was just open pollinated progeny collections from a seed orchard, that makes more sense, given that we do see a fair bit of pollen contamination in that orchard, apparently. So we have a question here for Tara from Antonio Castilla, asking about the ground penetrating radar you use to study root density. And could you maybe, do you think that this could be used in natural populations, or is sort of the homogeneity of a farm field site necessary?
I think that depending on where the trees were located in the natural population, you possibly might be able to use it in natural populations. However, I think there's a lot of concerns that you would have, such as how close the trees are together, what the ground is like, because everything's planted so neatly and in rows, it's easy to kind of distinguish when you're looking initially at the results, what belongs to what trees. I think you'd have a harder time teasing that out in natural populations as well. And then the feasibility of being able to bring the equipment out there and do all the staking to make sure you get good measurements. So depending on where it was, I think you can do it, but I think it works well in the orchard setting.
Great, thank you. I have a question for Dawei. So when I was watching this, I found your sort of transfer distance plots kind of interesting. One of the things that seems a little counterintuitive to me is it showed that lodgepole pine provenances from wetter provenances tend to perform better on drier sites. Is that accurate? Or sites drier than their provenance?
Like the optimum mean annual precipitation in the results is around 500 or 550 millimeters, and that is quite different comparing to other research. For example, in the BC, the lodgepole pine, the Illingworth, that one is the temperature. But in my results, I found that the precipitation is more like it's more associated with the height growth. And
yeah, that is quite interesting.
Yeah, that is quite different. And I'm also wondering, especially for the provenance trial, if you take a look at the map, like I do not have so many sites compared to the BC's Illingworth. Maybe that will be a reason. And other than that, maybe I will need like based on the current results I have, I don't think I'm able to explain why the precipitation affects more compared to the temperature. But for
yeah,
white
that
spruce,
is that is interesting.
Yeah.
Yeah, no, please continue.
Oh, what I was trying to say is that for white spruce, the result is more intuitive. It's based on the temperature.
Yes, that is very similar to what we found in BC. Let's see here. Got a let's see. Esteban, a question from Natalie Isabel. What do you think would be the minimum number of SNPs required to do this sort of seed lot monitoring? Because that's going to be very important for operational deployment.
Yes, and I just answered by typing, but now it is lost. I think that is in the answer one. So I said to Natalie, as I said, Jan Busquets' SNP chip worked so well with Alberta's spruce trees. I checked 2 SNP subsets with 2,000 SNPs randomly picked of 5,000 SNPs. So I wouldn't go below 2,000 SNPs for any calculations. Thanks for your question, Natalie.
Perfect, thank you. Tara, we have a, oh, it looks like you're already answering Keith's question in the chat there, but perhaps you'd just like to tell everyone here. Keith Jayawick-Ram has asked that lodgepole pine is a prolific seed producer in the wild, but seems to be problematic in seed orchards. Is that a fair assessment, and can you perhaps speculate on some reasons?
Yeah, so I was just typing an answer to this. So I think there's a variety of factors that they face while they're in the seed orchard versus in the wild. In the seed orchard that we're working on, it was established on previous agricultural land. So the land was originally used for crops. We're looking at different factors like the soil moisture, the nutrients. We're exploring the possibility that there's a hard pan layer so the roots can't properly colonize. And then we also have with the grafting process that when it's actually been grafted to trees that they spend excessive up to 2 years of time in pots. So then the roots could become pot bound and they're not able to properly colonize the soil, again, limiting the nutrients and the water available to the trees. So I think there's a lot of things going on with the trees when they're in the orchards versus when they're in the wild. That answers your question.
Yeah, no, that's very true. It may be of interest to you to reach out to some of the seed orchard operators and scientists in BC to sort of compare results and see, where there are maybe some similarities and differences between your two study systems, because those similarities may point you towards some causal factors there. We have a question for Dawei. Have you presented your results to tree breeders, and are they willing to use genomic tools for the design of seed zones?
Hi. So no, we haven't presented our results to tree breeders specifically. And this is the first conference we're presenting our results in so far. And I'm not sure if they're willing to use these tools, but based on my personal experience collecting phenotypic data, going on field trips and doing a lot of those labor work, I think it would be nice to have an alternative if it has a promising result. So yeah. And there's also another question for me in the feed loop from Barley Harris saying if I know this approach, the gradient forest model can be used in other conifer species. So I haven't worked on that. I have only worked on lodgepole pine so far, but I know there's other lab members working on other conifer species because we have the SNPs data for it. It's from the Aikens lab. And I hope it would transfer well based on the results we could see in lodgepole pine so far. It looks, the results looks quite good. And if it could be transferred to other conifer species, it would be a very good news, at least, I think. Yeah.
Yeah, and just to elaborate on that, I know lodgepole pine has fairly weak population structure. So basically your ability to produce what seem to be biologically and ecologically fairly reasonable seed zones from a species with such weak population structure does suggest that it should be applicable to other species.
Great.
Yeah, go ahead.
Oh no, I said great.
I have a quick follow-up for you, Dawei.
Yes.
Could you maybe elaborate a bit on the genetic offsets and how those might be applicable to seed transfer?
So for the genetic offsets we presented here in the conference is that we calculated the Euclidean distance between future climate and the current climate to see how vulnerable these species might be. But we haven't put that together with the zones that we predicted or with the existing reading zones. So maybe that could be another part to explore, and perhaps that would be that would provide some insight to see which seed zones will be more vulnerable. We did see, well, right now it's only visually saying these zones look like, for example, the zones that we predicted or the existing zones looks like it's in a vulnerable region in the genetic offset maps that I presented. But yeah, it would be better to quantify that as well.
Okay, great. Thank you very much. So that is going to conclude our time for this session. We still have some sort of open questions there, and I'm sure other people have more questions. Feel free to reach out to our speakers in the private chats. I'm sure they would be happy to speak with you more at length then. So thank you very much for attending, and enjoy the rest of the conference. And I think that's it.